Welcome to the Veterinary Rehabilitation Podcast, where we aim to help fellow veterinary rehab therapists increase their knowledge and elevate their practice. I'm Megan Kelly.
And I'm Ane Lloyd. Together, we bring you the latest insights, research, and information in the field of veterinary rehabilitation.
This podcast is brought to you by Onlinepethealth, a leading continuing education membership for veterinary rehabilitation therapists. With Onlinepethealth, you'll have access to a wide range of online resources to help you stay up to date with the latest techniques, advances, and trends in the industry.
Our podcast features in-depth conversations with leading experts in veterinary rehabilitation. They share their own experiences and knowledge to help you improve your practice. Whether you're a seasoned pro or just starting out in the field, our goal is to provide you with the tools and the insights you need to succeed.
So join us as we explore the exciting world of veterinary rehabilitation and help you take your practice to the next level. Hey vet rehabbers, the idea that tissues may possess some sort of memory is a controversial topic in manual medicine, and it's calling for research and some more clinical exploration. Many body workers at some point in their practice have experienced phenomena that may be interpreted as representing a release of memory traces when working on dysfunctional tissues. This feeling may have been accompanied by some type of sensory experience for the therapist or for the patient. In some cases, early traumatic experiences may be recalled. When this happens, the potency of the memory may be erased or eased, along with the restoration of the tissue function. Hence the question, can memories be held in the fascia, and are these memories accessible during manual fascial work? So before we head over to this exciting topic with Ane, if you're looking for some free online learning, in addition to our paid membership, which offers over 500 hours of vet rehab specific learning for equine, small animals, and hydrotherapy, we also provide free webinars in our limited access membership. The latest updates include core strengthening and lumbosacral stenosis, what can we learn from human back pain, lectured by Andrea Henderson, what's new in hydrotherapy research, lectured by Ariel Pichet-Markley, and managing the musculoskeletal health of the stabled horse by Gideon Tabor. Now, Onlinepethealth members, you can access these webinars in your membership, and for non-members, they're available at onlinepethealth.com/free. Now, if you have not yet bought your ticket for our annual online veterinary rehabilitation conference, the Vet Rehab Summit, please mark the date for the 31st of August online. We'll be delving into the ethical dilemmas that vet rehab therapists face, and you can find out more at vetrehabsummit.com. And don't forget, Onlinepethealth paid members, you get free VIP access to this event. Before we head over to this exciting topic that Ane is going to tackle, a quick word from our sponsor, Curacao Vet. Curacao is owned by Nada Robinson. Nada first developed her courses at the Colorado State University, where she started the Medical Acupuncture for Veterinarian Certificate program in 1998. And in so doing, she developed a science-based platform for veterinary acupuncture, the first of its kind. Today, Curacao Vet offers a comprehensive curriculum in integrative medicine with a rational, evidence-supported foundation. Courses include medical acupuncture, photomedicine, botanical medicine, medical massage, and integrative rehabilitation. You can discover Curacao Vet at curacao.org. Right, let's hear some more about fascia with Ane.
Good morning, vet rehabbers, and welcome to this month's Research Refresh. Today we are looking at, does fascia hold memories? And yes, I am still stuck on the subject of fascia. Even following the Vet Rehab Summit, I'm just continually fascinated by its different properties. As this article is not an open access article, I wanted to really share it with you guys in a research refresh, because I know that there aren't, not all of us would have been able to access this article to dive a little bit more into this holding of memories component of fascial properties. So let's dive into it. Right, their summary. The idea that tissues may possess some sort of memory is a controversial subject in manual medicine, calling for research and clinical exploration. Many body workers at some point in their practice have experienced phenomena that could be interpreted as representing a release of memory traces when working on dysfunctional tissue. This feeling may have been accompanied by some type of sensory experience for the therapist or for the patient. In some cases, an early traumatic experience could be recalled, and when this happens, the potency of the memory could be eased or erased, and there will be a restoration of tissue function that occurs. Hence, the question needs to be asked, can memories be held in the fascia? And secondary to that, are these memories accessible during manual fascial work? Okay, so let's discuss some of the ways that fascia could be holding memories. First, a neurofascial memory. Early investigation of fascia shows that it's well innervated, and we've continued to explore this, and we understand that fascia is very well innervated, especially by afferent nerve endings, so the nerve endings that are going back, sending signals back up to the central nervous system in the brain, and these include nociceptive nerve endings. Irritation of these primary afferent nociceptive fibers can initiate the release of neuropeptides, which in turn can alter the normal tissue texture of the surrounding connective tissue through their interaction with fibroblasts, mast cells, and immune cells. This process may trigger a number of local and global responses: connective tissue remodeling, inflammation, nervous system sensitization, pain, possibly evolving into persistent pain, and eventually followed by an adaptive response of the whole organism. Thus, under certain dysfunctional conditions, a neurofascial interaction may be responsible for the setting of a local tissue memory, or peripheral sensitization, followed by a corresponding spinal facilitation. This could lead to a global effect through the involvement of higher centers and of an autonomic and endocrine pathways. Changes not only of the neural pain signaling mechanism might follow in involving the function of somatosensory and cognitive affective areas in the brain, but fascial treatment may access such memories and obtain therapeutic effects. For example, in indirect types of fascial techniques, the unloading of the tissue can cause a consequent decrease of neural input and mechanical load through the fascial structures, possibly unloading muscle spindles while loading Golgi tendon organs. This may change the pattern of sensory input to the facilitated spinal cord area, quieting down the nociceptors. Local and spinal cord level autonomic reflexes could be stilled, particularly the sympathetic drive, which may have encouraged vasoconstriction and diminished lymphatic flow. This may encourage more normal pumping action of the muscles and an improved fascial sliding motion. So as we go through this, I hope that a lot of this biology, chemistry is already familiar to you. We already know how the nervous system is working. This is just another way of interpreting that information to help us understand a memory component of the tissue. So fascial memory. Memories in the body may also be encoded into the structure of fascia itself. Collagen is deposited along the lines of tension imposed or expressed in connective tissue at both molecular and macroscopic levels. So again, this is something we're familiar with, that collagen will be deposited along the lines of tension. Mechanical forces acting upon the internal or the external environment, such as posture, movement, and strains, dictate the sites where collagen is deposited. So, a tensional memory is created in a particular connective tissue architecture formed by oriented collagen fibers. This architecture changes accordingly to modification of habitual lines of tension, providing a possible medium-term memory of the forces imposed on that organism. This type of signaling may be altered in a pathological condition, such as locally decreased mobility due to injury or to pain. In cases of a functional strain or mechanical stress through collagen bundles, known as psychological responses involving fibroblast physiological responses involving fibroblast mechanochemical transduction and modulation of gene expression patterns, together with inflammatory and tissue remodeling processes of the collagenous matrix. Additionally, the release of substance P from nerve endings, particularly driven by hypothalamus following an emotional trauma, could alter the collagen structure into a specific hexagonal shape, which is referred to as an emotional scar. The entirety of this phenomenon may be interpreted as a highly structurally and functionally specific process of encoding memory traces in fascia. The extracellular matrix and tissue memory. In addition, this ability seems to be present not only in the collagen network, but also in elastin fibers in the various cells throughout connective tissue. This includes fibroblasts, mast cells, plasma cells, fat cells. Since these are relatively durable, long-lasting cells, they may represent a kind of long-term memory of the ground substance. The existence of a cellular network of fibroblasts within loose connective tissue may have considerable significance, as it may support yet unknown body-wide cellular signaling systems. The ground substance, in turn, provides a non-genetic memory complementary to the genetic one by ensuring a consistent set of signals to the cells. In fact, while genes may provide information to the ground substance on how to, the ground substance may define for the cell what to, shaping individual patterns of metabolism, development, growth, repair, and behavior. This microenvironmental memory underlying most pathobiochemical events has been indicated to be dependent on the matrix turnover and, as such, to be erasable via fibroblast induction and maintenance. The extracellular matrix could play a crucial role in sensing, integrating, and responding to the physical and chemical environmental information, either by directly connecting with the local adhesion or by regulating global cellular processes through growth factor receptor signaling pathways, which leads to the integration of both external and internal signals in space and time. Epigenetics and tissue memory. Epigenetics is the study of changes in gene expression or of cellular phenotype caused by mechanisms other than changes in the underlying DNA sequence. Epigenetic regulation of gene expression occurs as a result of alterations in chromatin proteins independently of the germline. This alteration of the chromatin architecture produces changes of the accessibility of genes and their expression that are preserved during cell division, and therefore they are heritable changes. In this sense, epigenetic modifications represent a kind of a family memory implicated in the control of several cellular processes, including differentiation, gene regulation, the development and genomic imprinting. Epigenetic changes, including DNA and histone methylation, might also cause a stable fibroblast activation, capable of altering immune function, producing inflammatory responses that may underlie the development of chronic disease. Epigenetic regulatory pathways may in turn explain differences in phenotypes between subgroups of patients and also between subsets of fibroblasts within an involved joint. As far as manual therapy goes, evidence suggests that mechanical signals are crucial regulators of cell behavior and tissue differentiation by affecting gene regulation at that epigenetic level through a heritable reduction of DNA methylation. This process may regulate extracellular matrix composition, inflammation, angiogenesis, and fibroblast activity involved in tissue repair and function. Microtubules, gel cell transformation, and tissue memory. Even subcomponents of the cytoskeleton, such as microtubules, seem to be capable of storing information and somehow being capable of retaining memory traces. Microtubules could actually act like little computers, storing memory in the form of information strings. They are polymers made up of polarized monomeric subunits known as tubulin, Information may be stored through different orientations of tubulin monomers. In addition, proteins like microtubule associated proteins may encode information to the microtubule by changing their position of attachment. For instance, the neural microtubule associated proteins have long been noted for their enhancement of tubulin assembly and microtubule stability, influencing the microtubule's spatial organization and interaction with cellular organelles, offering a record of cytoskeletal organization and cell function at the time of assembly. Interestingly, the information stored on a microtubule can be erased by depolymerizing it back into its monomeric units. Pressure and temperature change may cause these microtubules to depolymerize or to fall apart. Possibly this mechanism could underlie the changes in tissue viscoelastic properties as well as in the colloidal consistency of the ground substance after fascial treatment. Research suggests that an increase in the sliding of the tissue layers together with a decrease in pain following manual fascial work could be the result of a transformation of the ground substance from its densified state gel to a more fluid state gel sol. This change in viscosity seems to increase the production of hyaluronic acid together with the flow within the fascial tissue to improve drainage of inflammatory mediators and metabolic wastes, to decrease chemical irritation of the autonomic nervous system endings and the nociceptive stimuli to somatic endings, therefore resetting aberrant somatovisceral and or viscerosomatic reflexes. Manual therapy may then activate an erasing process via a gel-sol transformation of the matrix, causing a reset of dysfunctional memories, possibly stored in the fascia. So again, we're getting that gel-sol transformation through pressure and temperature changes. Fascial contractility and tissue memory. Another way in which fascia may encode memory traces is through its contractility and related reflexive spinal activity and central control. After the discovery of myofibroblasts in the connective tissue, the presence of an alpha smooth muscle actin in the structure of myofibroblasts has also been demonstrated, together with its responsiveness to mechanical stress. This supports a capacity of fascial contractility in a smooth muscle-like manner, and its potential influence on musculoskeletal dynamics as well as on resting muscle tone. The evidence of an intrafascial smooth muscle cell and autonomic nerves supports the hypothesis that a fascial pretension may be regulated via autonomic activity independently of muscular tone. Therefore, since fascia appears to be organized in tensile myofascial bands that comprise a single continuous structure, as we learned at the Vetriab Summit with the myofascial chains, the repercussion of an intrafascial restriction may be body-wide and may potentially create stress on any structures enveloped by or connected to fascia, which is all structures in our body. Dysfunctional memory might therefore be imprinted by the development of a fibrous infiltration and cross-linking between collagen fibers at the nodal points of fascial bands, together with the progressive loss of elastic properties. These changes in myofascial tissue could alter the activity of related superior centers, accounting for both sensory integration and motor control. A therapeutic touch might subsequently produce stimulation of pressure-sensitive mechanoreceptors in that fascial tissue, followed by a parasympathetic response. Under parasympathetic influence, a change in local vasodilation and tissue viscosity, together with a lowered tone of the intrafascial smooth muscle cells, could occur. And finally, in response to the proprioceptive inputs, the central nervous system can change its muscle tone, allowing the therapist to follow myofascial paths of least resistance to the point where correction of dysfunctional matrix cross-linking can be achieved. So I love that ability that that this system allows us to treat an area at a distant point to that area. When we have hypersensitization or acute pain in an area and our patients are not comfortable with us there, we can work in a distant area that's connected and have an effect, and I love that. The chemical memory. Another mechanism by which fascia could store messages is through chemical messages. A variety of substances are constantly transmitting innumerable messages throughout the body, including peptides, whose messages are relayed through receptors in target cells. Now, as I go through this, think about we are all familiar with the phenomenon where a certain smell can cause us to remember, can be linked to a certain memory. And this is how I understand that. So that chemical smell, that chemical molecule that is causing us to smell something has a link to a memory that is held in our body or in that organ where the chemical receptor is landing. Right, so let me keep reading, and then you'll see that connection with that understanding that we're all pretty familiar with. So depending on the precise external or internal stimulus, a particular information substance will flow through the body and bind to a specific receptor site. When this binding occurs, particular feelings are perceived, encoded with a given memory. Such chemical messages act reciprocally on the brain and the rest of the body, thanks to the denser presence of their receptors in the limbic or emotional portion of the brain. The body can therefore be conceived as a single organ with full sensing capabilities, where any tissue may store emotional memories based on the specific receptors they possess and the nature of the chemical messages that they receive. Manual fascial work may produce beneficial effects on chemically induced memory via a main pathway. The anandamide effect on the endocannabinoid system, the latter being an endorphin-like system constituted of cell membrane receptors and endogenous ligands. If you want to learn more about the endocannabinoid system and really focus in on that system in our bodies and how it works, Dr. Nada Robbins presented a webinar, which is in the Small Animal membership, on the endocannabinoid system. Go and have a watch. It's very interesting. This system influences fibroblast remodeling and may play a role in fascial reorganization, in diminishing nociception, reducing inflammation in myofascial tissues. Cannabinoids are also linked to cardiovascular changes, smooth muscle relaxation, and possibly mood changes through their role on the central nervous system. Fascial tissue work may also produce the enhancement of cytokine pools from actively proliferating fascial fibroblasts. Such fascia-derived cytokines may be delivered systemically, even in distant sites to that which is treated, via intrafascial blood flow, possibly reducing edema, increasing range of motion, decreasing pain, removing fibrotic materials, even at sites distant to where the manual treatment is applied. Tensegrity, vibration, and tissue memory. This is where I get excited. According to the tensegrity model, which we should be familiar with, the whole body is a three-dimensional viscoelastic matrix, balanced by an integral system of compression tensional forces in dynamic equilibrium. In this model, bones are the non-touching rods that play the role of compression struts, embedded in a continuous connecting system, or the tension system, that is the myofascioligamentous tissue in the body. Thanks to its hierarchical organization, any applied force can influence any part of the entire system, from cellular to the whole body and vice versa, via a non-linear distribution of forces, in such a way that local stimuli invariably leads to global reorganization. This structural and tensional continuity provides the basis for the continuum communication model. Cytoskeletons of epidermal vascular connective tissue and nerve cells, together with the extracellular matrix, forms an electromechanical semiconducting matrix that can generate and communicate coherent vibratory signals throughout the body. This system could be largely responsible for the rapid intercommunication that enables the body to function effectively as a coherent whole. It's been suggested that all major constituents of living organisms, including collagens and proteoglycans, may be liquid crystalline in form. This is important. In particular, fascia presents a crystalline collagen strands, with dielectric and electrical conductive properties that make them very sensitive to mechanical pressure, to electromagnetic fields, to changes in pH balance, and to their ionic composition. In fact, collagen fibers display a polarity within their molecular structure and can therefore generate piezoelectricity. The application of an electrical stimulus causes mechanical motion or vibration, while the application of physical force, tension, compression, or shear will generate electricity. Collagen fibers are arranged in highly ordered crystalline arrays, and this semiconducting system will produce coherent oscillations that move rapidly throughout the living matrix. Therefore, fascia seems to combine the property of a solid or liquid conductor and of a crystal generator system, which can generate and conduct direct currents as well as vibration. A consequence of such communication is that every process occurring anywhere in the organism produces a characteristic pattern of vibration that travel widely, distributing regulatory information. Every time a cell changes its shape or its metabolism, every time a muscle contracts, a neuron synapses, a gland secretes, the frequency of transmission of such vibratory signals changes throughout the body. The system is therefore instantly informed of events occurring everywhere in the network. Some theories hypothesize that the totality of such vibratory messages throughout the matrix may constitute a body consciousness, functionally interconnected with the brain consciousness of the nervous system via the crystalline liquid medium of the ground substance. Memories that relate to disease, dysfunction, pain, infection, injury, surgery, and physical and emotional trauma may be stored within the system, influencing the normal informational pattern either locally or globally, altering the properties of the fabric and distorting the resulting vibrations. In this way, consciousness may be influenced by memories stored in soft tissue. From a therapeutic perspective, it has been demonstrated that manual low-frequency oscillations can induce muscle relaxation, provoking a significant change in motor neuron excitability. In addition, both primary and secondary nerve endings exhibit sensitivity to vibration and sinusoidal oscillation, with a build-up in amplitude of response when oscillations near the resonant frequency are applied. So the amplitude of the oscillation and its frequency does matter. A slow rhythmic pendular swing has been shown to cause an inhibitory effect on the vestibular nuclei, resulting in muscle relaxation by inducing a psychogenic relaxation. And we're familiar with this, those of us who have kids and a swing. Vibration has been demonstrated to be a critical epigenetic factor on regulating the microenvironment of the extracellular matrix, thus providing a basis for reducing tissue adhesions and improving function. Finally, oscillations may promote intercompartmental fluid flow through a hydraulic mechanism, as well as having a possible modulating effect on spinal excitability and on the pain gate in the central nervous system. These beneficial aspects on the nervous system function following a vibratory treatment may also be due to an increase of the endoneural blood flow. Water and memory. Water molecules and their behavior in living tissues may also account for the ability of fascia to store memory. This part's really interesting. The collagen tissues of the body are embedded in layers of structured water molecules. Research suggests that interfascial water plays a key part in protein folding, the process necessary for cells to form their characteristic shape. When associated with protein structures, water can demonstrate a tendency to behave in a crystalline manner and to be influenced by properties that make up the cell, exhibiting structural organizations that differ from what is termed bulk water. The polarized water and ions surrounding proteins, including collagen, are highly ordered, forming chain-like filaments that hold the proteins together via hydrophilic interactions of hydrogen bonds. The system constitutes a hydrogen-bonded network that can support rapid jump conduction of protons much more rapidly than the conduction of electrical signals by the nerves. In other words, protein motions generate vibrational deformation of peptide bonds, which will involve polarization waves along the proteins, accompanied by proton conduction in the structured water shell. Thus, as a dielectric system, the organism may exhibit coherent vibrations as a result of metabolic pumping, where electromagnetic and electromechanical forces interact. Weak signals of mechanical pressure or electricity may therefore be readily amplified and propagated by a modulation of the proton currents or coherent polarization waves in the liquid crystal continuum. So, weak signals of mechanical pressure or electricity can be readily amplified and propagated. This system may account for a fast responding short-term memory of the ground substance, a memory that's dynamically distributed in the structured collagen-bound water network, self-reinforcing circuits of proton currents. Is that exciting? It is. Furthermore, liquid on its own includes coherent domains where all molecules oscillate in unison, in tune with a self-trapping electromagnetic field at a well-defined frequency. The coherent oscillations produce an ensemble of quasi-free electrons, able to collect noise energy from the environment and transform it into high-grade coherent energy in the form of electron vortices. This high-grade energy may activate the biomolecules resonating with water's coherence domains. So think about the advice that we got to use a tuning fork if we can't use needles to activate those treatment points within our patients. That's what's happening here. Therefore, flows of matter, energy, information, and memory can be spread and stored over the organism due to self-trapping, induced by the correlated coherence domains of interfascial water. Body workers can therefore gain their results by freeing tissue memories stored in the watery body content. The interplay of calcium ion concentration and unbound water oscillations may increase following manually applied fascial work, thus promoting interstitial fluid flow, and this may in turn stimulate fibroblast proliferation and collagen production or alignment, thanks to the coupling of the electrical vibrational continuum with the mechanical tensegrity structure of the connective tissue intracellular matrix. Boom. All right, so this was a lot of information, and I find it really interesting to take a different perspective and a different thought process on so many of the biological functions that we already understand and that we're familiar with, right? So what I want to take out of this is this little paragraph. It is therefore an informational network that distributes regulatory signals throughout the body, coordinating cellular and extracellular activities involved in growth, morphogenesis, and regeneration. When we when we think about the things that we've read now from a clinical point of view, what is really highlighted and very important to me would be hydration of the tissues, how we can use vibration and mechanical pressure and heat to influence the body and the tissues. Those are the things that I would be getting really excited about, that I am getting really excited about. So I'd love to hear from you guys. Any thoughts that you want to share, please get in touch with us. I really would love to hear from you and what you've taken out of this and what you're going to be thinking about further, learning about further, and potentially taking back to your patients. Have a great day, guys.
If you enjoyed this podcast, please hit the subscribe button so you get notified every time I load a new podcast. I'm here every week talking to vet rehab therapists from all over the world about all things vet rehab. A big thanks to our sponsor, Curacor. Their sponsorship allows us to be able to give this podcast to you for free. So please go and check them out at curacor.org. Don't forget to bookmark the next Vet Rehab Summit. It's on Saturday, the 31st of August, 2024. Come and be a part of the world's largest online veterinary rehabilitation conference created specifically for you, the vet rehabber community. Online Pet Health members get VIP complimentary access to the Vet Rehab Summit. For more information about continuing education for vet rehabbers, you can go to onlinepethealth.com.